Okay, so welcome to part 2 of this video on lipid rafts and cellular microdomains. Okay, so what we're now going to discuss is a very special type of lipid raft. So in the previous video what we discussed was that the cell membrane of cells is not just one homogeneous layer of, bilayer of phospholipids. Instead, what you have is you have regions where the makeup is very, very different, and these are known as lipid rafts. And instead of containing the normal phospholipids, instead they contain a lot of sphingolipids, specifically gangliosides, and a lot of cholesterol and ergosterol. Okay, so this is a lipid raft. And basically, these molecules don't just evenly distribute within the cell membrane. They all aggregate together and form a large collection of these different type of lipid molecules which sit together and form this lipid raft. Now, there are some very, very special types of lipid rafts known as caveolae. Okay, so there are some lipid rafts just look like this, but a special subset of lipid rafts, so I might put the symbol for subset, is the set of caveolae. So what do caveolae look like? Well, caveolae are special because they actually indent in, and what caveolae means is it's from some old language meaning little cave. And basically, they are these little rafts, these lipid rafts, which have invaginated into the cytoplasm like so. So when lipid rafts do that, when they invaginate into the cytoplasm, they are now known as a caveolae. Okay, now what causes lipid rafts to do this? Well, basically, it's a protein. We found proteins that cause this, and those proteins are called caveolins. So caveolins cause this. Right, and to understand how caveolins do this, we need to look at the structure of caveolins. For this, let's zoom in on the structure of this lipid raft. So you still have a bilayer of lipids in the lipid raft. Okay, the lipids aren't made up of phospholipids anymore, they're made up of sphingolipids and cholesterol and ergosterol, but you still have a bilayer, so you still have one layer like so, and another layer like this. Well, basically, so this is this caveolae, so I'll just highlight it pink to make sure everyone knows what I'm talking about. So these are the two layers of lipids that make up the lipid bilayer. Right, so these proteins known as caveolins, they have a structure like so. They're a polypeptide, they're a protein, and basically they go through the intracellular aspect of the lipid bilayer, but they don't make it through the extracellular layer, the outer layer of lipids. And then they curve back and then come out and go back into the cytoplasm. So this is the structure of a caveolin. And basically what these caveolins can do is they can bind to one another. So if I draw another caveolin in here, they will bind to one another. So they form a nice adhesion here, and then they can continue that. So another caveolin can come here, and that one can bind there. And this is how they start to form these caveoli, basically. Why they start to invaginate in this membrane. By them all connecting together, they they pull the membrane inwards, basically, and they form this invagination, this little cave or caveolae. Okay, so that's how these caveolins form, these caveolae, within lipid rafts. So often caveolae, often lipid rafts do form caveolae. They do associate with caveolins, and then those caveolins cause this invagination that leads to the formation of a caveolae. Okay, right, so now what we need to discuss is one other important aspect of how you make microdomains in cells, and that's the cytoskeleton. So the cytoskeleton is extremely important. Okay, so basically cell structure is maintained by the cytoskeleton. The phospholipid bilayer is a gooey layer, and if the cell was just made up of the cell membrane, it would have no permanent structure at all. You'd be able to mold it in any sort of shape you wanted. what holds it up is the cytoskeleton, which is a huge sort of protein meshwork that holds the cell together. And basically, if we imagine looking at the underside of this membrane, so let's imagine pulling out a sheet of membrane. So this is a sheet of cell membrane. So I've got a sheet of cell membrane, and we are now looking as though we're looking from the intracellular aspect. So we're looking at the intracellular aspect of this cell membrane. So basically, the lipid bilayer will consist of two sheets of lipids. So here is another sheet of lipid. So this makes up the cell membrane, and basically this is the extracellular aspect and this is the intracellular aspect. Well, basically, if you look at the intracellular aspect of the cell membrane, what you find is like a picket fence effect of the cytoskeleton. So you find that there are cytoskeletal proteins sort of like this. They sort of hold, they form a meshwork underneath the lipid layer, basically, this intracellular lipid layer. And that is what is holding the membrane up, basically. So if we were actually to look at this meshwork, if we took off the cell membrane and looked at the cytoskeletal meshwork, what we'd find is that it forms a great sort of sphere. So you have, loads of cytoskeletal proteins all underneath this lipid layer, this inner leaflet of the lipid bilayer, basically. And that's what is holding the membrane up. But this has another important function, because if I draw this picture bigger, the reason it's known as a picket fence effect is that, say you have these cytoskeletal proteins looking like so. Then, look, we've got this little region here. Now, these are big cytoskeletal proteins which are lying just underneath the inner leaflet, which means this is the inner leaflet of the lipid bilayer. So this is the inner leaflet, and this outer layer of lipids is known as the outer leaflet of the lipid bilayer. So outer leaflet. Right, okay. Then, if you have proteins sitting in this membrane, so let's say we have a protein here, which I could draw like this sort of protein in the cell membrane, then that protein has a portion of a domain, which sticks out from the inner leaflet and then extends into the cytoplasm. Well, basically, what we think of we think of proteins as being free to move around in the cell membrane. But what's going to happen is, if this protein moves around, it's going to bump in to these cytoskeletal proteins. It's going to get here, and then it's not going to be able to go past it unless this cytoplasmic domain, this portion of the protein that extends into the cytoplasm, unless that's very, very small, you're not actually going to be able to get past the cytoskeletal proteins. So this protein is effectively encaged or fenced in to this portion of the membrane, so it can't move. And that's why these are often thought of as being like picket fences. So this is the cytoskeletal proteins. So they're like fencing out the pen which this protein can sort of move around in, basically, cytoskeletal proteins. Okay, so that's another really important concept for making microdomains, basically. So, now let's discuss what the concept of a microdomain is, now that we've discussed lipid rafts and we've discussed this cytoskeletal protein structure. So basically, what you find is that adenylyl cyclase 8 and certain calcium channels are restricted to lipid rafts, and there is also cytoskeletal proteins helping them to remain in a microdomain. So, what you actually find is let's draw a picture here. So you find, let's say this is a cell, and let's say this portion here is a lipid raft. So you've got a lipid raft here. Basically, what you find in this lipid raft region is you find a lot of adenylyl cyclase 8 in this lipid raft region, and you don't find much adenylyl cyclase 8 outside of lipid rafts. And experiments have been done to try and determine, which portion of the adenylyl cyclase is actually necessary. Why does this adenylyl cyclase 8, and this is specifically adenylyl cyclase 8, why does this adenylyl cyclase 8 like to remain in these lipid rafts rather than being in the non-lipid raft portions of the phospholipid bilayer? Well, basically what they did is they gradually made mutations in this and saw how the distribution of adenylyl cyclase 8 in the membrane changes. And the bits that they found were absolutely essential for targeting adenylyl cyclase 8 to lipid rafts was not these two transmembrane domains. It was not TM1 and TM2. And that's surprising. You would think that it was going to be TM1 and TM2 because they're the bits which interact with the membrane the most. But it's actually this C1 portion and the C2 portion. And if you mutate those, if you take them out basically, then adenylyl cyclase 8 no longer remains in the lipid rafts and will be found in other portions of the membrane. So it's thought that these, it's not known how these achieve this, but it is known that they do achieve it, and it's therefore believed that actually they must be binding to other proteins that are targeted to lipid rafts, but as I say, that isn't fully understood. Right, now these adenylyl cyclase 8s have a very important role in non-excitable cells. So I need to talk to you about what a non-excitable cell is. So we have already talked about adenylyl cyclases in neurons and in heart cells, and how those adenylyl cyclases are influenced by calcium levels in neurons and heart cells. Now, these two types of cells will be counted as excitable cells because they are conducting action potentials continuously. Excitable cells. Non-excitable cells is the more bog-standard cell, let's say an epithelial cell or something along those lines, rather than one of these very, very special cells. Okay, so we are going to talk about what is the role of adenylyl cyclase 8, which is activated by calcium. What's the role of that being activated by calcium in non-excitable cells? And it's got a lot to do with these microdomains and these lipid rafts. Okay, so we saw in neurons how, and if you haven't watched these videos, I would highly recommend that you watch my video in which I'm talking about GI and auto-inhibition of neurons, and the video on how do beta-1 receptors cause an increase in the force of contraction or have a positive inotropic effect on cardiomyocytes. Those tackle the role of adenylyl cyclases and their interaction with calcium in these excitable cells. But we're now going to concentrate on non-excitable cells. And basically in non-excitable cells, what you find is that adenylyl cyclase 8 is localized to these lipid rafts. And you also find that other important proteins, which we're going to look at how these are important, but there's another very important protein that is found in here, and those are store-operated calcium channels, of which we've discussed briefly in the protein kinase C pathway videos, but we're going to discuss in more detail in upcoming videos. So this is a store-operated calcium entry channel, which has we'll discuss more what this channel actually is, but for now we'll just call it a store-operated. So store-operated. You will still see this terminology, but now the actual proteins that make these up have been identified and they've been given names, and we'll discuss all of that. Store-operated calcium entry channel. Okay, but often you will still see it referred to as the SOCE channel. Right, so you find these store-operated calcium entry channels also localized into these lipid rafts, and they basically stay within these lipid rafts. And what you find basically is that something about the lipid raft is keeping them in there. You'll also have this cytoskeletal contribution, so there might also be cytoskeleton skeletal proteins here and here acting as like picket fences through which these past which these cannot pass. But there's something definitely about the lipid raft there. Okay, you also find that the cytoskeletal proteins they're not just, you know, on the inner surface of the inner leaflet of the lipid bilayer. They also extend out inwards, and basically they demarcate like a small portion of the cell, which is going to be a microdomain. And basically, this is going to be in contact with the endoplasmic reticulum here. So you'll have the endoplasmic reticulum here, and basically you'll have a cytoskeletal protein sort of marking you off this little microdomain of the cell, where what's going to happen is all of this machinery is going to interact with the endoplasmic reticulum, and you're going to have cyclic AMP signaling in this little microdomain. And basically this is how you achieve a huge amount of specificity, how you can activate cyclic AMP, produce more cyclic AMP from this adenylyl cyclase 8, and trigger a very specific response. So this is a microdomain, because the cyclic AMP will just go up in this little sort of patch that you've demarcated. So I want to try and draw a three-dimensional picture of that. In fact, can I do it on this picture? So basically, you're not just going to have, you know, this in 3 dimensions. You need to imagine this is now in 3 dimensions. What you'll have is more like a cone of cytoskeletal proteins sort of demarcating. So this is supposed to sort of demonstrate that, you know, they'll go off like this, and they'll be marking out this cone, which is a whole 3-dimensional little region where this signaling pathway is going to act, and it's going to basically act with the endoplasmic reticulum, and we will look at this pathway in our video on calcium homeostasis in non-excitable cells. Okay, so that's the concept of a microdomain. Now I just want to briefly discuss a little experimental technique which can be used to show that these lipid rafts actually do exist, because there was a lot of contention about whether lipid rafts actually do exist. So we're going to discuss a technique known as fluorescence recovery after photo bleaching. Okay, so fluorescence recovery, or FRAT for short, fluorescence recovery after photo bleaching. So this is one of these techniques that was used to try and assess whether adenylyl cyclase 8 was really co-localized in these lipid rafts, and whether lipid rafts actually did exist. Fluorescent recovery after photo bleaching. Okay, so the idea basically is that you can tag certain molecules with fluorescent markers. So for instance, we could tag the molecule ergosterol with a fluorescent marker. So let's say we have tagged ergosterol with a fluorescent marker. Ergosterol with a fluorescent marker. Okay, well actually, this first experiment, this isn't going to be fluorescence recovery after photo bleaching, but I'll just show you. Green fluorescence protein. So this is an experiment that would back up that the existence of these, of these lipid rafts. So basically, you could tag ergosterol with green fluorescence protein. And then if you looked at the distribution of ergosterol in the membrane by looking at the activation of this green fluorescence protein. So basically, what will happen is, you know, the ergosterol will be distributed in the cell membrane, and as we've discussed, it will mainly be in these lipid rafts. Now, when you shine UV on green fluorescence protein, it starts shining green light back out at you. So you shine in UV light, and it shines out, it emits visible light in the green portion of the visible spectrum, visible green. Okay, so basically you can see where the ergosterol is because you can see the green light. So you find large green patches within the cell membrane that look like this. So that's a nice piece of evidence for the existence of these lipid patches. But that's not fluorescence recovery after photo-bleaching. Let's now see how we can see that these lipid rafts actually do have some sort of different kinetics, basically. So let's tag adenylyl cyclase 8 with green fluorescence protein. So we can tag adenylyl cyclase 8 with our green fluorescence protein here. And basically, again, what we'll see is that it will be localized to these lipid rafts. But basically, what we can also do, and actually that's not fluorescence recovery after photobleaching either, but what we can do to try and understand how these lipid rafts are different from non-lipid rafts is that we can take a protein that is expressed in both this is finally fluorescence recovery after photobleaching. We can take a protein that is expressed in non-lipid rafts and in lipid rafts. So let's say we've got some protein that is expressed in both of them, and then we can tag that with green fluorescence protein. Now, what we'll see is when we look at the entire cell, we'll see green everywhere, basically, because this protein is expressed everywhere. Okay, right, now what we can do is we can photo bleach, which basically means we shine very, very intense UV light onto our protein, and basically it destroys the fluorescence, it destroys the green fluorescence protein. The green fluorescence protein receives too much energy and it destroys it. So that's bleaching our green fluorescence protein, so that's the photo bleaching portion. So if we photo bleach certain portions of the membrane, then what will happen is the instant we do it, what we'll find is that, let's say we photo bleach this portion here, we'll find that it no longer fluoresces green again. Now, usually what happens is that the green fluorescence will recover, and the reason is that this protein will diffuse from neighbouring portions of the membrane, and it will sort of all mix up, and you'll get your green fluorescence in this portion of the membrane recovered. So that's why it's called fluorescence recovery after photobleaching, FRAP for short, because we are looking at how the fluorescence recovers. Okay, so if you do this in the normal part of the membrane, the non-lipid raft portion, you get a pretty quick recovery. The protein sort of redistributes and you get it back, and it returns to where it was before, basically. But if you do it in lipid rafts, what you find that the recovery time takes a huge amount of time, and basically it's because the movement of proteins through lipid rafts is much, much slower than the movement of proteins through non-lipid rafts, because the viscosity of the of the lipids which make up the lipid rafts is much higher than the viscosity of the phospholipids which make up the non-lipid rafts. So basically, recovery after photobleaching, fluorescence recovery after photobleaching, takes a lot longer in lipid rafts. So if you did do this for adenylyl cyclase 8 tagged to GFP, then you'd find that recovery of the green fluorescence once you photobleach, so let's say you photobleach here, then the green fluorescence is going to go there. Well, it's the recovery, the time for it to return there, i.e. the time for AC8 with green fluorescence protein to diffuse from the neighbouring portions will take a lot longer than it would if you were in non-lipid raft regions.